welcome learners and viewers in general. So, this part of the program we will try to link with the previous program that we had on the DNA modification or DNA methylation, and in this program we will try to link DNA methylation to the cell differentiation, that is to suggest that the DNA methylation or DNA modification through methylation has an impact, has a correlation with the expression of that gene, the gene which has undergone methylation. So, as you have said that not all genes are active at all time. This is one of the strategies also of the cell to conserve energy, that all the products of all genes are not needed all the time. The products which are required for those genes only need to be active, and in order to save energy of the cell, rest of the genes whose products are not required, they need to be shut down so that we can conserve energy. So, here also this phenomenon is more pronounced in bacterial and prokaryotic cells compared to eukaryotic cells. Now, we will discuss a small, very straightforward experiment now, which gave us a clue, a window to suggest that these methylations are in fact associated to differentiation. See, the whole life sciences research today is engaged in explaining the mode of differentiation, and if we can understand differentiation, perhaps we can also understand de-differentiation. So, the main focus is on cell differentiation. In this regard, an experiment was conducted by McGee and Ginder, which have utilized a unique property of a chemical compound called 5-azacitidine. These two scientists compared the methylation status of the beta globin locus in cells that did and did not express this gene. Using restriction enzymes that distinguished between methylated and unmethylated DNA, they showed that beta globin locus, this is important, beta globin locus or gene was essentially unmethylated in the cells that were producing beta globin, and in cells where this beta globin protein was not present, and when that gene was examined, it was found that that was methylated. We have already discussed this, that the methylation leads to compaction of the nucleosomes, and demethylation leads to relaxation of the DNA. This was the observation, and then coupled with this, that 5-azacitidine experiment, what they did was utilizing the property of 5-azacitidine, because it is one of the many chemical analogues for the nucleoside cytidine. When these analogues are integrated into growing DNA strands, 5-azacitidine severely inhibited the action of DNA methyltransferase enzymes. DNMTs their ability was severely damaged by 5-azacitidine. That normally methylate DNA, these methyltransferases were almost rendered inactive. Most DNA methylation was known to occur on cytosine residues of CpG. We know it. These are CpG islands, cytosine, phosphate, guanine island. It was hypothesized. The premise of their experiment was that if they inhibited methylation by flooding cellular DNA with 5-azacitidine, which is an analog, then they could compare cells before and after treatment to see what impact the loss of methylation had on the gene expression. As I told you, knowing that gene expression changes are responsible for cellular differentiation, these researchers used changes in cellular phenotypes. What was the parameter? When they cultured cell lines, so any change in the phenotype of the cell was taken as an expression of gene expression, as a proxy for gene expression in the cells. Say small data that they obtained. They also ran a control to show the effect of cytidine analogues on cell differentiation and DNA methylation. See, these are three simple columns, chemicals added, 3 micromolar cytidine, which was a control, number of differentiated cells, 0, there was no differentiation, and amount of methylation was 100%. So what do you infer from this? In this control, no differentiation was seen, whereas 100% methylation was seen. That means methylation has suppressed the differentiation. In another set of sample was 0.3 micromole arabinose cytosine, which is another analog of cytosine and also acted almost like a control. Here also no differentiation and 127% methylation was measured. Now why 127? That shows that there were some cytosines other than CPG islands which also got methylated. Whereas the experiment containing 3 micromole of 5-azacitidine showed remarkably different results compared to control and aracid side. That 22,141 cells were found to be differentiating. That means they showed some differences in their morphology, and it was taken as the initiation of differentiation. And if you compare it with the methylation pattern, we can see that only 33% of the CPG islands were methylated. Now it suggests that methylation has a direct correlation with the cell differentiation. And after this experiment, actually, they opened up a window for the world scientists to work on these lines, making these as a premise that the DNA methylation is linked with the gene expression. It was published in Jones and Taylor in 1980. So these experiments opened the door for investigators to better understand exactly how methylation impacts gene expression and cellular differentiation. Evidence of this has been found in studies that show that methylation near gene promoters varies considerably depending on cell type, with more methylation of promoters correlating with low or no transcription. Now, a very classic study was conducted in the population of bee. In a bee colony, there are 3 individuals: drone, worker bee, and queen bee. All these 3 individuals have the same genetic makeup, and yet they have remarkably different functions in a beehive. This is a classic example. It's a very, very landmark paper which says the honeybee epigenomes, differential methylation of brain DNA in queens and workers. I want your attention to be drawn on to this heat map. These are the heat map of worker bee. These are mRNAs, the worker bee and queen and drone. So heat maps of drone and worker show some similarity to some extent. You can see that major portion of the worker and the drone is red, but it is the queen whose heat map is entirely different from worker and drone. So this is nothing but they have linked the functions of these individuals to the methylation pattern of the DNA. You see these green and red structures are nothing but the extent of methylation. Red is 100% methylated and the green is 0% methylation. So this is also evident here that most of the DNA of queen is showing green portions, which is unmethylated or hypermethylated, which is expressing itself more than the drone and workers. These are some of the biosynthetic pathways which are also affected by epigenetic regulated phenomenon, and these are cellular differentiation. Totipotent cells become pluripotent. That is also, you see, the stem cell, the embryonic stem cell retains the entire ability of developing into an organism. But as it evolves and becomes multicellular structure, it loses its property of totipotency. We can explain it now that as it is dividing 1 into 2, zygote becomes blastola, gastrula, the DNA is getting methylated. The DNA is getting methylated, and what triggers that which portion has to become euchromatic and which portion has to become heterochromatic? That trigger, that environmental or nutritional or what triggers the expression of genes has not been understood very well, but we know this much, that the cells keep losing their potential as they evolve and as they grow and as they differentiate. Therefore, a stem cell can give rise to a stem cell, but after it has divided into various other components, it cannot be reverted back to the totipotent stem cell. X chromosome inactivation, as you can see here, is the Barr body. Gene expression of one of the female X chromosome is downregulated, which becomes an isolated chromosome and becomes a Barr body, and DNA methylation, and it is the expression of DNA methylation and histone modification. Imprinting is epigenetic marking of a locus on the basis of parental origin that results in monoallelic gene expression. You see, in the diploid organisms, there are two alleles, one is recessive and the other one is dominant. But here, the gene expression takes place on the basis of only one allele, the other allele becomes silent, and that is because of the differential methylation or DNA modification. Now, there are methods to sequence DNA, that is the dideoxy chain termination method is there, Sanger's methods are there, and now they have been evolved into a next generation or capillary DNA sequencing. But how to study the sequences which are methylated? There are a number of methods, but the only reliable method is the method of bisulfide deep DNA sequencing, or we can call it bisulfide conversion. For the benefit of those students who are having a chemistry background, they will understand that the basic premise of this experiment is that the bisulfite modification technique uses bisulfite salt to oxidatively deaminate cytosine residues on single stranded DNA, converting them into uracil. That is, cytosine gets converted into uracil. The unmethylated cytosine gets converted into uracil, and because of the oxidative deamination. But the methylated cytosines, they remain intact. They still have got their methyl group attached. So, in three different steps of sequences, we can find out the pattern of DNA methylation. You see here it is unmethylated DNA, methylated DNA. Original sequence is this, where the C, not all C, but this C, because of the CPG island, it is methylated, CPG, methylated, CPG, methylated here. So, bisulfite converts cytosine into uracil, and when we do PCR, that is polymerase chain reaction, uracil is converted into thymine, not converted actually, it base pairs with the thymine during replication. So, by sequencing them again, we can differentiate the methylated cytosine and unmethylated cytosine. So, this bisulfide sequencing is the only reliable method to find out the methylation pattern of the DNA. So, we have understood that genetics and epigenetics are different in the sense that in the genetics the DNA sequence alters, whereas in epigenetics the DNA sequence remains the same. It is only certain nucleotides get additional functional groups, and they make an impact on the gene expression. We have also seen that DNA methylation is linked with the disastrous consequences. It is also linked with the gene expression, and DNA methylation and unmethylation, they are also linked with the gene expression, and we can study methylation pattern through bisulfide deep sequencing.